Let's talk about active transport. Everything, again, we talked about diffusion. We talked about facilitated diffusion. We talked about osmosis. Each of those three things does not require your cells to use up any of its ATP, any of its energy. So if it had a preference, that's what it wants to do. Okay, it wants to use one of those three methods to get material across the cell. If it has to, it'll use a second method. If it's not passive, it's active. And in this case, the word active gives you a clue. This is going to require energy to do. This is going to force your cells to use up some of its energy. It doesn't want to. It wants to save that. But if it has to, it can use its ATP to do these. Here's why. The reason it has to use up energy is because we're going backwards. We're not going from high to low. We're going from low to high. Okay, the comparison I would like to put out there for you, to help you remember this a little bit. If you've ever gone canoeing. No? Yes?
Yes.
Okay, all right. Maybe just not. Maybe you haven't done that.
I think one.
All right, so since we live in Brookville, Indiana, we live right there on the Whitewater River, and there's a whole bunch of different canoe places that you can go. Okay, there's Morgan's, there's whitewater rafting, there's several different places.
I like going whitewater rafting.
If you haven't gone there, you might stop in. If you have ever gone canoeing, if you haven't gone canoeing, I'm sure you've gone biking. Thank you. If you've tried to paddle upriver, or if you've gone biking and tried to go up the hill, it stinks. Okay? It takes a lot more energy to go the opposite direction. Same thing with your cell. If I've got a whole bunch of stuff on the outside, it's going to take a lot of energy to try to push things into that. Okay? It wants to come in, and we're forcing it to go out. Low to high. It's reverse. So we say it's against the gradient. We're not going down the hill this time, which is easy to do. Right? Sledding down a hill is fun.
I sled up hills.
To walk uphill or sled uphill
That's what I did.
takes a lot of energy. You can sled uphill. You just have to paddle yourself up the hill.
No, I just
That's not
fly
sledding.
right up the hill.
That's like snow.
That's working hard, okay? Nobody wants to do that. All right, so when Benjamin goes down the hill, it's great, but then he complains because he has to walk
That's
up
the worst
the
part.
hill. I mean, it's like, oh my gosh, it's so fun, and then you got to walk back
Is
up.
Benjamin your son?
What if you don't
Yeah,
like the
Benjamin's fine.
Paris place?
Mine's fine.
Or you could go to, yeah, you could just rig up a place that they pull you back up. Yeah, so like the commercial places, they just hold on to a tow rope. Yeah.
I don't think our cells have one of those.
Well, our cells don't have that. So. Okay,
I'm sorry, I didn't know that.
let's look at the first example. All right. First example, active transport, called the sodium-potassium pump. This is actually an integral protein. It's one of those carrier proteins that's going to be using a lot of energy, and this happens inside your nerve cells. So your brain, your spinal cord, your nervous system. It's a really good example of this. It's called a sodium-potassium pump, and the way it works, got a little description there. 3 sodium ions are being pumped out, in other words, moved. We're going to move 3 sodiums out and bring 2 potassiums in. And this is actually vital to your nervous system working properly. We're pumping sodiums out and potassiums in.
Was it a 2K what?
2 potassiums pumped in.
Oh, okay.
Okay. So we're going to kick. We're kicking sodiums out, 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 out. Okay.
Out.
And then we're letting the potassiums come back in. Well, over time, you're going to end up with a cell. Here's our cell. If we keep pushing the calciums out, you're going to have lots and lots and lots and lots and lots and lots of calciums outside the cell. Okay, where does the calcium want to go? Man, it wants to go in. Okay, it's like pushing more and more and more water on the other side. Okay, it wants to come in, so we're pushing calcium where it doesn't want to be. That takes energy. All right, let's take a look at an animation.
Okay, great.
Everybody got that written down? Good? Okay,
Wait, what
take
is
a
the
look.
3 not?
3 sodiums.
Is that a plus sign?
Yeah?
Okay.
Because sodium's a cation.
I'll just
Okay.
make sure I wrote it right.
All right.
The sodium-potassium pump is an active transport
This
mechanism.
one's fancy.
Let's
3
watch it.
sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient.
What would cause this process to stop?
Whenever it's even.
All right. 1, Clayton said lack of energy. Well, if we go back to this step,
So,
boom, what if there wasn't a little ATP? There wasn't energy.
would they just...
It would just stay like that. Okay, it would never change shape. It'd never kick those out. The whole process, all right, stops. All right, no ATP would stop this. What else might stop this?
What if potassium is in
Okay, in this particular shape, it doesn't really show that very well, Whitney, but imagine those aren't there.
Okay, it's only...
Okay, in this particular form, it's really attractive to the sodiums. Okay, so they're going to fill in those spaces. Imagine that's not there though. Okay, now when the ATP binds to it, it changes its shape, and what you should know from proteins is their shape determines their job. So if it changes shape, boom, it's got a different job now. All right, so now imagine these circles disappear, so it doesn't attach very well anymore, but these will appear. Okay, so now in that shape, it's really attractive to the potassiums, which can lock on. Okay. So if there's no sodium, would this happen? No. If there was no potassium, this wouldn't happen. Okay, but it's moving them the opposite way they want to go. Okay, that's like trying to cram more and more people into a crowded elevator. Nobody wants to be there, it's all crowded. Okay. Question there. Last one. So we've got sodium potassium pump. That requires energy. Second one that requires energy is called endocytosis and exocytosis. These are opposites of each other. Endo is bringing materials into the cell. And exo, think of exit. We're getting rid of materials, moving them out of the cell. Okay, endo, into, exo, think of exit. Okay, exoskeletons on the outside. Okay, we're moving things out of the cell. And these are basically the same process, just one's bringing it in and one's bringing it out. So you just kind of have to really remember just one of the processes and you've got both. Okay, everybody got that written down? I'll show you what this looks like.
No. Alright, yeah, I'm done.
Now, the key word is bulk. All right, we're bringing big amounts. Okay, we're moving the semi-truck all the way into the store instead of just unloading the whole thing. Piece at a time. We're going to bring the whole thing in. Let's see if I can find it. Can't find it. I'll have to click it again.
What?
Maybe. Maybe too big. I don't think you guys can see it. Move it there.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism.
So here comes a
Phospholipid
white
released
blood
by
cell.
mammalian cells,
There's a
or by
bacteria.
components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide,
Don't
and
worry about all
complement
these phrases.
receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b
Think of those
receptors on
peripheral
phagocytes.
proteins. Things
This
can attach
process
to
of
it.
coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome.
Okay. Now, what you just saw there, let me kind of go backwards. Zoom. So there's a bacteria. It could be a virus even. And your white blood cells have the ability to engulf things. Okay, that takes a lot of energy. Think back to when you guys were playing that cell game. Okay, whenever you moved that cell, you had to use up ATP. You had to use up energy. Okay, that takes a lot of energy to do that. Zoom. Okay, once it's inside,
Lysosomes fuse with the
Take some
phagocyte,
lysosomes.
releasing digestive
gonna
enzymes
blast
such
it
as
apart.
lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents
Here's
of the
exocytosis.
phagolysosome are eliminated from the phagocyte by exocytosis.
okay, there we go. All right, so exocytosis is basically it's taking out the garbage. So we've got some sort of vesicle. It moves closer, closer, closer. Dumps whatever it is outside the cell. Big amounts. Okay, so you don't have to take a little bit at a time. Okay, bless you. All right, questions on that?